Hello everyone. Today we are going to learn a prokaryotic cell structure and its functions. Uh, Today I'll tell you the overview of the prokaryotic cells and their functions. Topic number 1: executive overview of the prokaryotic cells. Let me read first. Prokaryotic cell represent one of the most fundamental architectural design of life on the earth, characterized by the absence of well-defined membrane-bound organelles and membrane-bound nucleus. These organisms are exceptionally efficient, versatile, and structurally streamlined, while the prokaryotic exhibit an immense variety of microscopic shapes, size, and biochemical configuration. Their fundamental and internal organization remains remarkably unified and consistent across the taxa. Here, the prokaryotic cells represent the most fundamental architectural design on the life on the earth. This means that prokaryotic cells are simple as compared to eukaryotic cells. They have absence of well-defined membrane-bound nucleus, membrane-bound organelles. These organisms are fundamentally similar, but they have primitive nucleus, primitive main membrane. They have absence of membrane-bound organelles, etc. Now, here the taxa represent the category. Taxa represent the category. So, key core characteristics of the prokaryotic cells. Key core characteristics: well-defined nucleus is absent in prokaryotes. Nuclear envelope means the boundary around the nucleus, that we call the nuclear envelope membrane, absent. Membrane bound organelles means the organelles which has membrane on as its boundary, that is called membrane bound organelles. Organelles are the distinct means structure which are scattered in the cytoplasm. Membrane bound organelles are absent. And ribosomes is in universal, it is found in all means cells. So ribosome present as a 70 s particles. 70 s particles means the 70 is it is the unit. 70 years it is the unit, and it further divide into 50 s and 30 subunits. Means yes means it was discovered by scientist Swedberg. You will study in detail afterwards, but till here you can see that. And representative organisms, bacteria means the organism which represent or which belong to prokaryotic cells or prokaryotic organisms, that are bacteria, cyanobacteria, Bruggen, algae, and mycoplasma, and pleuropneumonia like organism. Pleuropneumonia means very small organisms. So the next topic is the complex cell and envelope and its modification. Means cell envelope means the cell which has boundary, that is called envelope or membrane, or we can say it is also known as lamella. Most prokaryotic cells, particularly bacteria, possess chemically intricate, tightly bound three-layer structure of protective cell envelope. Each layer possesses a distinct biochemically specialized properties, yet they coordinate such as functional unit. Means complex envelope means such as this is cell, and this is nucleus, and this is cytoplasm, and these are some ribosomes and some organelles. Means if suppose this is the cell wall, sorry, if suppose this is the cell wall, so this cell wall consists of this this cell wall consists of three layer structure, that is glycocalyx, plasma membrane, and cell wall. Cell wall is present in plant cell and prokaryotes, but plants cell wall is absent in uh, animal cell. And cell wall is the unique character of that what we can say animal cells, plant cells, sorry. So let us discuss about that three layers. Layer position, we can see here the outermost layer, we can see structural entity, that is glycolics. Glyco means glucose and calyx means coat. We can say here calyx means coat. So now biochemical composition and core function differs widely in composition and thickness among the various bacteria. It can be manifest as a loose sheet, gelatinous sheet called slim layer. If it is a slim layer, means what we can say, it is thick, means thin and loose sheet, it is called a slim layer. As it is thick and tough and highly structured configuration known as the capsule, it facilitates the surface attachment and defenses against the host immune system. I think this language is too easy, so you can understand this. Now moving to the next part, that is middle layer is cell wall followed by cell wall. This is in sequence, sequence. These layers are in sequence. So let us discuss about the cell wall, a rigid structure that defenses the exact shape of cell and delivers robust structure support to prevent the osmotic bursting or mechanical collapsing. uh, Means it is a rigid structure and it is also non-living. Deliver the stropage, means it also gives support and prevent the cell from undesirable molecules and bacteria, prevent from infections, etc. Now let's see the examples. Bacteria found uh, predominantly from peptide peptidoglycan, means the cell wall of the bacteria are made up of peptidoglycan. Archaebacteria composed of pseudomurine, means their what we can say, composed of, means their cell wall is composed of pseudomurine. That what we can say, the cell wall which the organism which does not possess cell wall, that is exception for the prokaryotic cell, that is mycoplasma, which do not have cell wall. And the size, and what we can say, the size of this mycoplasma is about 0.3 micrometer, means its length, and its size about 0.1 micrometer. Now moving to the next layer, that is innermost layer, that is cell membrane. As we know that cell membrane is selectively permeable. That selective permeable means that can what we can say, it is able to interact with outside world. Okay, now let us understand its function and how it is composed of. A universal, highly flexible, selectively permeable lipid bilayer. It is formed by lipid bilayer, and major lipids are phospholipids. phospholipids uh, Regulates the molecular traffic into out of the cell, interacting dynamically with the external environment. Its structural design is also fundamentally analogous to the eukaryotes. Means now you can understand this. So here we have finished that three layers. Now moving to the next, that is genomic and extra chromosomal genetic architecture. uh, I think this is not concept based, but we can understand that genomic means it related to genetics, and chromosome means genetic material. Now, prokaryotes maintain uniquely streamlined genetic architecture consisting of the two distinct structural components: the primary nucleoid region, autonomous extrachromosomal plasmid. Plasmid means small DNA, small circular neck DNA. Now, nucleoid means the what we can say, the structure which is present, means which is present In the nucleus, the primary genetic material is necked and lacking the shielding nuclear envelope or histone complex protein. It lacks. One second. Let me. Huh. Which lack the nuclear and histone is the protein which is present in the chromatin fiber. It is generally structured as the single highly folded double-stranded circular DNA molecule. Instead of basic histone protein, the negatively charged genomic DNA is structurally bound by the positively charged polyamine proteins, which is made up of polyamine proteins carrying the NH3 plus functional grouping. NH3 is we can say carbon compound. It is the functional group as amino group to safely pack the chromosomes within the dense uh, cytoplasm. Now moving to next part, that is plasmids. Plasmids are the specialized extra chromosomal small double stranded circular DNA which are capable of independent replication. They can replicate independently without any help. They replicate autonomously without needing permission from primary nuclear genome. Plasmid are not essential for basic cell survival and not means phenotypic, means it is the phenotypic character of the prokaryotic organisms. Now antibiotic resistance, plasmid also are resistant to uh, antibiotics. Antibiotic resist the harbor genes that actively neutralize the antibiotic agent, frequently changing membrane structure and locking the channel to prevent the antibiotic entry. Gene taxis is gene vectors are also known as gene taxis. Plasmids serve as a natural vehicle to transport specific genetic sequence between bacterial during conjugation or molecular engineering. I think that is not part of the syllabus, so we will discuss afterwards. uh, internal cytoplasmic modification and uh, mesosomes are the specialized cells which are formed by the extension of the plasma membrane. They significantly increase the metabolic surface area, enzymatic concentration of the cell, organized into three distinct forms: tubules, vesicles, and lamellae. Lamellae is also known as membrane. Their diverse function includes tubules Tubules mediate DNA replication and equal distribution of the daughter chromosomes, help in DNA replication. Vesicle facilitate the cell wall synthesis and active around metabolic protein secretion. Lamellae essential for cellular respiration and energy production. Character respiratory chain enzymes, means lamellae is also called as membrane. Okay. Chromatophores are the pores which are found in photosynthetic uh, uh, prokaryotes like cyanobacteria. These are inward extension of plasma membrane, means here the light trapping pigments are present such as chlorophyll. They are directly responsible for executing the photosynthesis. Inclusion bodies and gas vacuole. Inclusion bodies are the non-membrane bound completely neck, and these what we can say, these serve as the disposal of the reserve materials in prokaryotic cells. It is the character of prokaryotic cell. These are lie in free in the cytoplasm. Phosphate granules accumulate and store the inorganic phosphate. Glycogen granules act as a primary reservoirs of carbohydrate energy. Synophysian granules store specific protein and pigments useful for the photosynthetic pathway. Gas vacuoles, unique character found in blue green pulper photosynthetic bacteria. They show metabolic gas, regulate buoyancy, help in cell position itself and perfectly within the column for ideal light absorption. So now moving to the next topic, that is surface appendages and mortality. I think that surface means these are motile structure, locomotory organ, we can say locomotory organs or structures. uh, One thing I missed that to tell you, that is motile. Motile means it is the capacity of a organism to move independently. Dependently, independently. Bacterial cell display specialized surface configuration extending outward the govern of the locomotion and physical interaction. Surface appendages, these are the outgrowth of, one second, let me change the color. Outgrowth outgrowth of cell membrane. Flagella. Flagella is Flagella is long, thin, filamentous protein structure extending outward from the cell wall. They are composed of three explicit segments, that is, filament, hook, and basal body. They are sole structure responsible for cell active motility. Pili. Pili are elongated tubular configuration constructed from specialized protein. Pili are what we can say constructed from special protein, that is pilin protein. They not play a role in motility but are crucial for conjugation and mating. Long tubular structure that helps to monitor the DNA of one bacterium to another bacterium, or transfer of DNA from one bacterium to another bacterium, or food, or anything. That is fimbriae. Fimbriae, small rigid bristle like what we can say fiber sprouting out the uh, actively out the cell surface, the primarily to attach the bacteria from tightly environment surfaces such as rock and streams. These these pili and fimbriae are surface structures but do not these are surface structure but do not play role in motility. motility Now let's try some questions to uh, analyze our knowledge. Now I'll ask, you can see 6 question, high yield assessment and critical review questions. These are NCRT based and line by line questions, so please don't skip them. 1. The chemically complex cell envelope of prokaryote consists of sequentially 3 which layers? We have studied that 3 layers: glycolysis, cell wall, and cell membrane. That is correct answer. Now, the next primary genetic material of prokaryotes is technically referred to as the second one is correct answer. Now, polysome, I think I have not taught you, but polysome is a chain where the ribosome attached to single mRNA chain and form single strand of mRNA, means chain, what we can say. Yeah. Now third question, 4. uh, mm, One second. huh which of the following biological process is not the recognized as the functional role of mesosome? Functional: cellular respiration, ATP generation. Second, genomic DNA and हे बरोबर आहे. आणि cell wall synthesis and active protein secretion. Direct active. This is not the function of mesosome. What are the precise structural sedimentation coefficient of prokaryotic subunits? Means I have taught you this one, 70 units, and eukaryotics 80 units. Now last question, moving to the last question, that is gas vacuole, which provide the essentially buoyancy control and found in which organism? I have taught you blue, green, purple, and green photosynthetic area. So here we stop our discussion, and thank you for attending my lecture. And if you want like this more, subscribe for more, and comment me which chapter you want next. Thank you all of you.